this is one of our SEBA program, which is SEBA Expert Seminar Series. So we regularly have this seminar throughout the year. So this time around, we have our guest speaker from Nara Institute of Science and Technology, Dr. Minotsu. She is currently working in the, if I'm not mistaken, the plant symbiotic lab in NICE. Is it correct? Yes, and under the tutelage of Prof. Dr. Satuko Ishida. Right. Right. So today, the topic would be about cyst nematodes, which is what Dr. Minotsu is working on, worked on, and let me first introduce her. Assistant Professor Dr. Minotsu currently is a JST Presto researcher and assistant professor in NICE, Nara Institute of Science and Technology in Nara, Japan. She completed her postgraduate, master's and PhD of agriculture from Nagoya University, and after completing her PhD, she had the opportunity to do a postdoctoral work in John Innes Center, United Kingdom, for 4 years, and then she came back. I believe you came back and joined NICE afterwards, yeah. So her research interest focuses on plant microbe interaction, especially between plant and fungi and plant parasitic nematodes. And right now in NICE, she is also venturing into the field of parasitic plants. Yeah. All right. So I'm really excited to have Dr. Minotsu today to talk about her work on cyst nematode. And without further ado, I would invite Dr. Otsu to present her work. So I would like to pass the virtual space to you whenever you are ready.
Yeah, I'm ready. So I'll share my screen first. So and start my presentation. Okay, so can you see my slide? Okay, so yes. Okay, so thanks for your kind introduction. So I'm Mina in Japan, and thanks for giving me such a great opportunity. So it is a great honor for me. And as he said, I just came back from the UK last year, and now I'm in Nara. So I briefly introduce about Nara. Yeah, Nara and NICE. So the NICE, the full name is Nara Institute of Science and Technology. Sorry, I haven't full name in NICE. So anyway, so the NICE is a graduate school. It is not a normal university. So the NICE is a graduate school and just has the master course and the doctor course. So quite so specific for the research, and it is very highly NICE. So there are many NICE researchers in NICE, and NICE is here. So next to Kyoto and Osaka. So Nara is not so big city, but it is very convenient to go to the big cities. And Nara is not big cities, but very famous in Japan as well, because here, this is a map of the shrine, and this is just part of the shrine in Nara, and there are so many old temples and shrines in Nara, because Nara is the oldest city in Japan, and it was an ancient capital in Japan. Of course, now capital is Kyoto anyways, Tokyo anyway. So, and this is a picture of the big park, including many temples, and deer are walking around everywhere in this park, and very cute and very lovely. So, and this is one of most famous large statue of Buddha, and it is very huge and around 10 meters, and surprisingly, it was made more than 1,000 years ago. So the Nara is very nice city. So if you come to Japan, so just go to the Nara Park or somewhere in Nara. So I can guide or something. So just let me know. Okay, so let's move to my seminar. So now, as he introduced, today's talk is the infected cell induced by the cyst nematode named Syncytia, and I focus on the structure of Syncytia. So before talking about the 3D structure, I briefly introduce about the plant parasitic nematode. So the plant parasitic nematodes are a kind of plant parasite, as the name suggests, and they cause really big problem in the agriculture, and crops and vegetables were lost around 157 billion every year because of this kind of nematode. And there are big three of plant parasitic nematode: root node nematode, cyst nematode, and root region nematode. So, they are very, very similar. And if I mix all of them at the same time, I think you guys cannot distinguish which is which, I think. But they are completely different species, and life cycle and disease symptom induced by each nematode is completely different. This is a root node induced by root node nematode, and this is a shrinked root induced by root region nematode, and they were cysts of cyst nematode. And among them, I focus on this cyst nematode species because the system to induce infected cell by the cyst nematode is unique. The cyst nematode induce cell fusion in plant cell to form big syncytium. So I'd like to introduce more how cyst nematode infect plant tissue and how induce syncytium in plant cell. So after invading into host plant root, nematodes walk around toward to the vascular tissue to get the nutrient, and then they decide the infection site and pierce their stylet into the host plant tissues. The stylet is this straw-like structure and a kind of the nematode. And Japanese. So the nematode secrete effector from the gland via the stylet and modify cell wall, plasma membrane, and plasmodesmata, like this. And eventually, around more than 30 host root tissues were fused and form a big syncytium, like this. However, the mechanism of cell fusion in plant is little known, so that is why the syncytium formation mechanism is wrapped in the mysteries. So in this study, I keen to understand how cyst nematode induce cell fusion in plant cell and how form syncytium in plant tissues. And I believe this study helps crop protection in the world and reveals cell fusion mechanism in plant. However, there are some difficulties to investigate syncytium formation because the infection process are completely inside the root like this, and it is very hard to observe without any sectioning. That is why, so far, the observation using sectioning are generally used for the syncytium formation studies. Of course, the sectioning is quite useful and I also use that, but the bisection observation, the information of real infection process and the 3D structure of syncytium are completely lost. So in this study, I tried to establish new imaging techniques to elucidate the syncytium formation induced by cyst nematode. And in this talk, as I said, I just focus on the 3D structure of syncytium. And for investigate the 3D structure of syncytium, I use the soybean cyst nematode, HG, because it has been in Japan very long time ago, and it is very easy to handle in Japan. The problem is the host plant, because I want to do the imaging, but as you know, the soybean has very fatty and thick roots, and it is not so good for the imaging. So that is why I try to screen many legume plant. And finally, I found this Chinese milk veg, the Astragalus cynicus, is the best host for the imaging among the soybean cyst nematode. So I use this combination to do the 3D imaging. in today's talk. And before showing some 3D images, firstly, I'd like to explain a bit what happened in syncytial cell during the infections. So this is a section picture of early stage of syncytium. This is an electron microscopic picture, and red shows the nematode, and yellow shows the syncytium. And this is a high magnification picture of this area, and blue shows the cell wall. And here, cell wall were partially dissolved and cell fusion were occurred here. But it is very difficult to know. This is very nice picture and this is beautiful picture, but it is very difficult. It is very time consuming work. You need to make 1,000 sections and preparing the electron microscopic observation and checking everything under the microscope. So it is very nice, but yeah, bit difficult to do that, especially lazy person. So I am lazy person. So so that is why I try to establish the 3D imaging system by using the whole tissues. So this is very easy. I just infect nematode on the plant and just I fix and clearing, and I stain by using the chemical dye and using the confocal or two-photon microscopy to observe. And the upper panel shows the uninfected root, and the bottom panel shows the infected root. So here, about the cell wall, if nothing happened at the cell wall were equally stained blue like this. However, when nematode infected in the root, as you can see here, the high magnification picture is here. So there are some holes on the cell wall. And also, so we could see some enlarged nuclei in the syncytial cell. So by using this method, we could see the morphological changes of nuclei and cell wall without any sectioning and without any electron microscope. And I think it is very useful to check briefly under the microscope, I think. After you get very nice picture, and then you do maybe the electron microscope or something. That is the easiest way, I think. So I checked my method is good for here I checked my method is good for the nematological work. And let's move to the latest stage of the infection. And this is a section picture of the 21 day post-infection, and the yellow dot shows the syncytium here. And these black bar are the breaking down cell wall. Of course, from this picture, we could find something happened in the syncytial cell wall, but it is very difficult to guess and difficult to know what is happened in syncytial cell wall in detail. So I do the 3D imaging of the syncytial cell wall during the later stage. And so this is the uninfected root. And as you can see here, the cell wall were equally stained. On the other hand, this is the infected root. Here is a nematode, and here is a syncytium. And as you can see here, compared with the uninfected root, there are some weed pillars in the cell wall. And by closing up and 3D constructing here, it looks the Parthenon in the Greece. So from this data, we found there are some pattern for the breaking down cell wall in Syncytia, and it is not random. Also from this picture, the fluorescent intensity of pillar looks stronger than other place and in the cell wall in infected tissue. So intensity level of pillars were investigated. So as you can see here, the colors in pillar are white to red, the high. So it means the highly fluorescent compared with other regions. And the bottom graph shows the quantified intensity, and it obviously revealed that pillars were stained strongly than other regions here or here. So the cell wall staining chemicals, calicofluor white, directly bind the cell wall in the cellulose in the cell wall. And this suggests that pillar containing more cellulose than other cell walls, right? So this indicates that the cyst nematode regulate both cell wall synthesis at the pillar place and the degradation at nothing place to form the Parthenon pillar in the syncytium formation. And previous studies showed that the soybean cyst nematode induced syncytia some region in plant root, for example, in the cortex and in the stem. However, there is no information about the 3D structure of each syncytia. So next I investigate if there are any morphological changes, morphological difference between them depending on the positions. To checking this, I just compare the cell wall morphology between the cortex and stem syncytia. And the pillars like cell wall, which I shown you before, were seen only in the cortex. So this one. But we want to know, so which side can we see in this kind of pillars? So I made a video and check each side if I can see pillars or not. And this video shows the blue side cell wall here. As you can see here, so there are some pillar-like structures. But this is the left side of cell wall here. So you can see some dot-like structure, dot-like cell wall, but we couldn't see any pillars-like structure here. So this is a green side of cell wall here. So we could see something happen, but still nothing, still no pillars in this side. So from this video observation, the pillars were only from the typical side of the cell wall, and nematode tightly regulate cell wall remodelings like this. And next, I check the morphology of the syncytium in the stem layers like this. And also I check every side of the cell wall. And this is a summary picture of them. So I checked every side, but I couldn't see any pillar-like structure in any side. So these results show that cell wall pattern were completely different depending on the positions. But so here, the plate-like cell wall were observed only in the blue panel, not in the red and green panels. So this indicates that this syncytium in stem layers also has some direction, like a syncytium in the cortex as well. So both syncytial cell wall has special direction something. So, and this is a summary of my talk. So during the early stage, so 2 days post-infection, I could see the enlarged nuclei and partially broken down cell walls without any sectioning and without electron microscope. And 14-day post-infection, that is a later stage, I could find there are two types of the syncytia depending on the positions. So when nematode may induce the syncytium in the cortex layer, the cell wall like structure and syncytia expand toward to the xylem and phloem to get nutrient. On the other hand, if the syncytia is in the stem, the cell wall was plate-like patterning, and the syncytia expand along with xylem and phloem like this. So this is the first time to see the 3D structure and first time to found the 3D, the difference between the cortex and cell layer syncytium, I think. And now, of course, I think how Cyst nematode induces such a complex 3D structures in the host plant tissues. And there are some special tissues in plant as well, such as phloem and xylem, because the phloem has some seed pore at the same side of cell wall. Also, has very special cell walls, like a ring. So from these tissues, I think the cyst nematode hijacks some mechanism in host plants, such as phloem and xylem formation to, of course, not only xylem and phloem formation, but the cyst nematode hijack many mechanisms to induce the syncytium, I think. And to understand the syncytium formation in detail, now I try to establish a new experimental system for live imaging, because previously I didn't talk today, but I, of course, tried to do the live imaging, and finally I found the chemical dye to staining living nematode, and also I established the transient transformation system of the Astragalus cynicus and did the live imaging like this, but still very difficult because there is no stable transformation system for both, and it is very difficult to functional analysis. So now I try to use another nematode and another host something to investigate how this nematode form the syncytium. And this is my lab, previous and current lab. In this nematode study, I was starting at my PhD in Nagoya University, and during my postdoc, I stopped to do the nematode work. And from last year, when I back to Japan, I restarting the nematode work here. And she is Satoko Yoshida, and Satoko is working about plant parasitic plant, as he said, as he introduced. So I am the only person who working plant parasitic nematode in this lab, but if you are interested in the parasitic plant or plant parasitic nematode, please come and join us. Thank you for the listening.
All right, thank you very much, Dr. Mina. It's very interesting to see the images, like really distinct changes that occurred due to the cyst nematode infection. All right, so now it's 11:23. We have time for Q&A session. So I'm sure a lot of the audience have questions to ask. So if anyone wants to ask question, you can use the reaction button, hence the raise hand function, and introduce yourself before you pose your question to Dr. Mina. So I invite everyone. Okay, Dr. Sharifah, maybe you can have the first question.
Thank you, Dr. Fikri. Hi, Dr. Mina. I'm Sharifah. I'm working on Melodoja incognita on banana plants. Looking at your new interesting but lazy system way of I understand the laziness because all this while I need to be chopping off and doing cross section of the roots. But do you have any specific characteristics of root system to work on in order for you to get this 3D view, or can we just work on any type of root system, for example bananas? But
yeah, I think yes, because that is I think you can buy it.
I can just buy it?
I think so. So this clearing solution is very special, and my colleague was established and my colleague was made, but now I think you can get it because some company were offered to him to maybe share the protocol or something, and I think you can order it. But that is
order the solution calfluor.
No, the calfluor is the cell wall staining solution, and you need to order the clearing solution, so clear C. I can maybe we're here.
Later on, maybe we can just I can just contact you to get
Yeah.
I'm interested to find out how the root knot nematode develop the galls in root system.
And I
that
think
hasn't been done.
not my study, but some other group working on the root knot nematode also do the clearing tissue by using this kind of clearing solution. So I think maybe it is really helpful for you, I think, as well, I think. So anyway, so just maybe email me. I can share the information about it as well. So.
And I don't understand at the point where you've got different concentration of the fluors when you stain it after 2 days of infection in the syncytium, whereby you've got different colors intensity of the fluors. What does it mean?
So you
when
mean
you
this
see
one?
that? Yes. What does it mean?
Pillars.
Yes.
So this charcoal white will directly bind to the cellulose at the cell wall. That is why the stained more mean there are many cellulose there. That is why the fluorescence intensity higher than other lesions.
Because you said
Meaning
the stain
dissolution
binds to cellulose. Yeah.
The dissolution of cell wall would mean increased number of cell walls in the area of syncytium.
So here the remain cell wall.
Can you make it bigger, Dr. Mina?
Put it on slideshow, please.
Sorry, so what?
Can you put it on slideshow maybe?
Yeah, okay. No. This one?
Or different view, yeah. Yep.
Sorry,
So Dr. Pa is asking why you have like different concentration of the cellulose. I think because this stain, this cut off, what was it?
Cellulose.
You mentioned that it binds cellulose, right? So this shows the intense the level of cellulose accumulation in the pillars, right?
Yes.
I see. So that signifies cell wall dissolution. Okay.
Yeah. So I just used maybe only one concentration of the chemical staining dye, and just I stain the staining, the depth is different. That's it. So depending on the cell wall content.
All right. Thank you, Dr. Mina.
Yeah. Thank you.
Thank you so much. We have another question from Dr. Ardiana. Go ahead, Dr. Ardiana.
Hi, hello, Dr. Mina.
Hello.
Very nice presentation, and I did my PhD at NICE as well with Demura Sensei, working on cell wall. So your presentation really excites me. The structure, the pillar structure, and also the other types of structure, kind of like remind me the formation of xylem in the roots. And perhaps you know that for xylem, there are different patterns, like for example, for protoxylem and also metaxylem. And you mentioned that the different patterns of the syncytiums depends on the position where the nematode infects. So you're talking about, like, we understand imaging is not easy, and we need to have a good system, good interaction between the host and also the pathogen, but as well of the samples itself. So because I'm also curious about the pattern, why did the nematode hijack and induce different pattern? How does this pattern would contribute to the the propagation of the nematode itself in the plants? And have you considered of using a cell suspension? I don't know, because if, let's say, the nematode hijack and produce a factor molecules that allow the host to modify its own cell wall. So I don't know whether you can see such pattern modified in the cell suspension system, which is easier to observe.
But I haven't tried, but I like to try in the future, but I think it is very difficult. So, but yeah, that if I can use the cell suspension, that is perfect. But yeah, let me see.
Thank you.
Yeah.
All right, we're open. Thank you very much, Ariana. We have time for more questions from the audience. Perhaps I can put one in. I'm curious about the method that you use, the photon microscopy, if I'm not mistaken.
Yeah,
So is
two-photon
this
microscope.
method different from other methods? Is this more advantageous for the purpose of this experiment?
Yes.
Or
Yes.
because I'm not really
yeah.
familiar with the technology.
So no, I didn't describe the in detail. So here, the two-photon microscope is the special microscope. Is maybe its two-photon microscope is better for the deep tissue imaging compared with the normal confocal microscope. So the laser can reach to more deeper region than other microscope. That is why I use that. So
I see.
yeah.
But how about the staining? The staining molecule, it has to be specific to this microscopy, or is the same?
No. So this cytox orange and calicophore white is maybe very universal one, and you can use for other microscope, not only the confocal and two-photon, also other maybe normal fluorescent microscope as well. But I think if you want to see in detail about the morphological changes in plant tissue, something, I think maybe you need to use the confocal microscope, something, I think.
So for this experiment, you had to excise the root and then look under the microscope. Is that correct? Or you can you use like live samples that like you uproot the plants and then view under the microscope and then report the plant?
No,
Is it possible?
So basically, for the imaging, I growing plant on just media.
I see.
Yeah, and the plant is not so big. I think this kind big. So.
Oh yeah. So I saw the picture. You used a different I can't remember the species name, but you decided on that particular plant because of the thin layer of root. Is that some type of lotus plant? Because I saw the flower is.
The kind of, but not lotus japonica something.
I see. So it's like it grows on soil, it's not in like a water.
Yeah, I just use the image something. Yeah.
Aquatic plants. I see. Okay. All right. Any other questions from the floor? I have many questions, but I would like to allow other people to ask questions. Any questions? Please you can use the raise hand button, yeah, if you have a question. So just okay. I think I can ask more questions. Regarding the pillars, so it's just rearrangement of the cell wall, if I imagine. So for me, I imagine the cell wall to be like a one flat surface, and then through the infection, it causes like the gaps to occur vertically in the wall. Is it correct? Because you mentioned that it involves dissolution and synthesis. So if through my imagination, it's like you have a complete wall, a flat wall, and then the nematode induce the formation of the gaps without causing new formation of the wall. But you mentioned it's synthesis. So can you explain more about that?
So because I haven't, this is not my study, but some other study also checked another cell wall component. And in the syncytial cell walls has very unique cell wall material such as arabinan or something, so which is normally not in the cell wall. So that is why the nematode synthesize not only normal cell walls, also another maybe unique sugar.
I see.
At the cell
So
wall.
not commonly found
Yeah.
in the plant, but by the infection, you start to detect those.
Yes.
I see.
Especially around the pillar like structures. Yeah.
Because very unique that you see this kind of structure. Is it is the pillars like hollow? It's like it's not hollow, like it doesn't have any space in the pillar, right? It's like solid
Yes,
structure.
Yes. Probably I
I see.
haven't cutting down the pillar, so I don't know.
Yeah, because I was thinking whether it's connecting between the different cells or it's just only in the syncytium.
I think only in the syncytium, but it is very difficult to distinguish which is the end of the syncytium something. So I think maybe I think
it's
we
very
need
interesting to see that it has a specific orientation because it's only forming up vertically. It's not cross cross-linking.
Yeah.
So it's really specific. So yeah. And then is yes,
Sorry,
Dr.
Dr. Fikri. I think maybe you would also be interested to see whether if there's lignin also deposited together with cellulose, because the pattern very similar to xylem. So
but that is the typical primary cell wall component. So I think so. In previous study, checking the component in this kind of cell wall, so there is no typical secondary cell wall component.
So only for primary cell.
Yeah. But yeah, of course, I haven't checked the lignin yet, so it is worth to try. Thank you.
Dr. Sharifah, tadi I saw your hand raised. You have a follow-up question?
I had a question, but I forgot. It just came in the middle. I lost it.
Too many things running.
But maybe just to throw some ideas out. What I'm interested in is to actually look at what are the proteins in the syncytium or the galls after looking at the formation of them. So the problem normally is once we stain it and that's it, we can't use that particular sample anymore. So is there any way that you can see it and then use the same route and then get the proteins or RNA out and do some molecular work on it?
I think the problem is fixing. I think if you didn't do any fixing step and just staining something, I think you can use the tissue for the RNA sample something. Yeah, just.
But then the problem when you fix it and that's it, right? Once you fixed it, you can't use this route anymore, right?
Yes, but if you didn't do any fixing, you can use that one for another the experiment, right? So
but staining would change would do something to the cells, correct? Staining, if you stain it.
I think it depends, because if you just want to stain nuclear and cell wall, I think you can still see something. Didn't disappear.
So did you?
Right?
I've got the question. The question comes already. So did you slice? Did you slice the roots and put it under the microscope, like to make it thin? Then you see all these, or you just use the whole root system?
Yeah, in this case, I just use the whole root. Yeah.
So you did not compress it, and that's why you see that.
Yeah, I didn't compress anything.
Oh, this is good technology then.
Yeah, that is why I talked about it.
I will definitely contact you.
So you have the microscope in NICE, this two-photon microscope? Or was
two-photon microscopy, no. So because I was used that one in Nagoya University, but I can use that one, as well.
I see. So you have to go to Nagoya if you want to do this kind of experiment.
But there are many nice confocal microscope in NICE as well. So I think we can use that one as well, because in this case I use two-photon microscope, have to do both live and fixed imaging. But I think about the fixed imaging, I can use the confocal as well.
Confocal here.
Yeah.
Okay. Maybe you can update us with another session if you with the results that you have from the confocal later,
Yeah, I
whether it
think
works or not. Right. Okay. So, do we have other questions from the floor? No. Maybe one last question from me. So, is this formation of this specific formation of structure, this pillar structure, is it has been observed elsewhere in different condition, in different tissue, in any different situation
so
in
you
plant
mean
tissue?
maybe during plant developmental step, something.
I'm not really sure. Like, is this a specific change that occurred by induced by the nematode, or is like a similar process?
I think so. I haven't seen anything. I haven't seen this kind of pillar-like structure during the plant developmental stage, something. So the most similar pattern is the xylem, but not completely similar, but yeah. But this is very weak and this is very unique. So
because I think, is there any other pathogen that can induce syncytium formation? And maybe it's this kind of pathogen also produce this kind of changes in the root system and the tissue in the tissue.
I think so. So some pathogen also make some goal, but the strategy is different, I think.
Yeah,
so,
I think it's different.
But so I think no one checking the cell wall status during the pathogen infection or something. So it is possible there are some similarity between the some fungal pathogen infection and nematode infection, but yeah, we need to compare them, I think.
And you think if cyst nematode infecting different plant hosts, they can also induce the same changes in the root tissue, like forming these pillars in different plants as well?
So now I am having look because it is possible the Astragalus cynicus root has this kind of potential to make this kind of pillars. So I think it is possible it is the same between the plant tissue, but it is a little bit different, especially the like this. Yeah, the still layer.
It will be interesting.
Yeah.
So I hope you can update us with
Can I ask one more
details.
question?
Yeah, sure.
Did you extend your work on until the nematode has a full cycle, life cycle completed in the root? Did you extend to see how the syncytium actually helps? What's the final form of the syncytium?
I think
whether it's maintained.
This is the mostly final form. I think this is the mature stage, and this is the male nematode.
Male.
Yeah.
Oh, the small one there, right? Or the whole thing?
What? Sorry.
The whole broken lines is the nematode, or the small thread there?
This is this white line is a nematode, the main nematode, and this is a part of the syncytium. So, and this nematode is already in male stage, so that is almost final stage of the syncytium, I think.
What about the female?
So, in
would
this
female induce different type of, because it's the female that we care about, right? I didn't know that the males actually produce syncytium.
Because about the cyst nematode, the half of them are not half, but mostly half of nematode are male and half of the female, not the same as the root nematode. Yeah.
So meaning even the males are damaging too, is a concern. The males are our concern too. Infecting, I mean, producing syncytium and affecting the roots.
Yeah, both can infect and make syncytium.
Okay.
Yeah, because both sex need to the reproduction. In case
so
of the
both
system.
male. So this is parthenogenesis life cycle. Meaning the males are also producing eggs.
No, the male and female are needed to the reproduction. I think the root node nematode, female is only need for the reproduction, right?
Yes.
But about the cyst nematode, so not only female, also male are needed because they need sex. And
because what I thought, what I understood was, the male would go out of the root system, and then the sexual reproduction would happen outside.
Step. But that is final step. After the maturation, they of course moving out to the host root, but during the J2 and J3 and J4 stage, they need to make the syncytium and get the nutrient from the host.
I see. Okay, that's quite different. Then I'm interested to also see how how when they produce eggs, and how would the syncytium actually develop in the vicinity of the eggs, how until it hatches. Do you have any research done on that?
I haven't checked that stage. I checked just maybe matured female or something, but I haven't checked any egg or something. But I think the syncytial status is not so different between the male and between female and the egg frying
would it
female.
somehow become dormant at one point during the whole life cycle? Do you expect that to happen?
Sorry, what? So during the life
Dormant
cycle?
as in there's not much activities occurring in the syncytium, like it will die off at one point?
I think it will die off. So I think this 14 to 16 days are maximum of the syncytium. So I think it is important to see here and not so important maybe more later stage, I think.
I see. Okay. All right. So that's just the reason why I thought maybe I can see the figure before non-infected 2 days up to 14, and then after that what's happening to it. Okay, thank you, Dr. Mina, for entertaining me.
Thank you, Dr. Thank you, Dr. Sharifah, for the questions. I'm sure Dr. Mina appreciate the questions. All right, I think that's about it. That's about the time we have for today. I thank everyone for joining this session, and most of all, I thank Dr. Mina Otsu from NICE for her time and her her presentation. It's very, very interesting. And I would like to remind all the participants to please look at the chat box and fill in the survey box, a survey form for this program. We appreciate your feedback lah, and just fill up a simple feedback form, and it will only take several minutes. Thank you very much. And again, I hope everyone enjoyed and enjoyed the session we had today. And with that, I would like to close the session lah. Thank you very much, everyone, and see you next year with our new 2013 sessions of SEBA's Expert Seminar Series. All right. Thank you so much, everyone. Bye-bye.